Cancer sucks. We all know that. One of the most exciting fields in biostat research is figuring out how to kick cancer in the nuts. Thanks to advances in genetic screening, we can identify cancers down to specific mutations and create new therapies that are hyper-specific to different cancers. This spares our healthy cells from the scorched earth of traditional chemotherapy. But pharma companies have a big problem. Tuning therapies at the gene level can potentially create thousands of new drugs to test, but because ethics exist, we can't just give them out like candy. Therapies need to be extensively tested via clinical trials. The traditional approach in clinical trials can be thought of as one drug, one disease. This has worked in the past, but it's expensive and can take several years before a therapy can be accepted and brought to market. When there are thousands of potentially life-saving therapies waiting in line, pharma companies need a solution to evaluate all of them fast. And statisticians have solutions, and we'll discuss one of them in this video. This is PocketStat, a series of short explainer videos for concepts in statistics and biostats. In this video, we cover the basket trial. A basket trial is a modern clinical trial design which allows us to evaluate a single therapy in multiple diseases. It's most commonly used in cancer. Often, cancers in different areas of the body will have the same genetic mutation. If we create a therapy that can target this mutation, in theory, it can be used on all of these different cancers. But cells are complicated and other factors get in the way. The therapy might be effective in a few of these different subtypes, or even just one. We refer to these different cancers as baskets. To start a basket trial, we identify a few cancers with a common genetic mutation. The disease doesn't have to be cancer, but there needs to be some unifying aspect that therapy can take advantage of. Then, we recruit people to these baskets, give them the therapy, and see if some of them have a favorable response. This is usually something binary like remission or the shrinkage of a tumor. But where do the interesting statistics come in? To understand this, we need to understand the classic statistical approach. In each of these baskets, there's some number of people and some subset of them will have a response. What statisticians are interested in is estimating the therapy's response rate, or the proportion of people who will respond favorably to the treatment. If it's high enough, then we can push this therapy to larger confirmatory trials. One way to estimate the response rate is to do an independent analysis, where the data for each group is used alone to estimate a rate for each basket. One weakness of this approach is that it's vulnerable to small sample sizes. This is especially true with cancers, since some can be very rare. Small sample sizes usually result in bad estimation. The other extreme is a pooled approach. Here, the data from all baskets is put together to estimate an overall response rate. This overcomes the sample size issue, but there's a serious assumption that comes with this approach. Pooling will average the response rates over all of the baskets. This is fine when the response rates among the baskets are all about the same, but that's not usually the case. Consider a scenario where the therapy will truly work in only one basket. This is usually called a nugget scenario. The true response rate of this nugget will be considerably higher than the other baskets. But if we pool the data together, the estimated response rate of this nugget will be dragged down. There's an increased chance we might make a mistake. We might accidentally conclude that the therapy will be ineffective in this nugget group, a type 2 error. To understand how a statistician might think of this, think of pooling and independent analysis as two extremes on the spectrum. Pooling shares the data entirely, while independent analysis has absolutely no sharing. The innovation is in the middle of this continuum, a partial pooling approach. What if we shared data only between baskets that seem like they respond similarly to the therapy? If we can figure out how to share data just between these similar baskets, then we can get the benefit of both worlds, an increased sample size and better estimation of each basket's response rate. But these benefits come with a new statistical problem. How do you actually decide which baskets to share between? In other words, how should I, the statistician, model this sharing? One of the earliest approaches is what's called a hierarchical model. In a hierarchical model, we suppose that there is some overall or population level response rate in these baskets to the therapy. The response rate of an individual basket can be thought of as a small shift from this overall mean. When we consider the response rates of all baskets, we can assume that they follow some distribution around this mean. We call it hierarchical because there's two levels here. The first level is the population distribution, where the basket response rates come from. The second level are the actual responses we see in the people in these baskets. Assuming this hierarchical structure gives us partial pooling, and the level of pooling is decided by the variance of the first level distribution. When this variance approaches 0, we get back the pooled approach, and when it approaches infinity, we get back the independent approach. This isn't the only way, and it won't be the best way in all situations. Statisticians have created many different ways to produce different sharing mechanisms between the baskets. targets. And this research still continues today. It's one of the ways statisticians contribute to getting more therapies into the hands of those that really need it. This was a Pocket Stat on the basket trial. If you want more content like this, consider subscribing. If you'd like me to make a particular video about a concept, let me know in the comments. Thanks for watching, and I'll see you in the next one.